Hello everybody and welcome back to my YouTube channel. And in today's video, we will be discussing about top 25 MCQs from microbiology. So, when I talk about microbiology, it is a very deep subject. इससे mostly there are domains from which the questions could pop up. The number one domain is sterilization. So, from sterilization, you can expect a huge amount of questions. And the next domain is from bacteria, virus. So, from bacteria and virus, either from their structure or something else. The third domain is from staining. So, gram staining, acid fast staining, IMVIC staining. From those particular points, there could be question. The fourth domain is source of antibiotics and the different microorganisms used in the assay, microbiological assay, like E. coli is used in chloramphenicol, and sources streptomyces gracius is a source for streptomycin and all. So all this is very important when I talk about microbiology MCQs for GPAT. So the first question is which of the following is not a sterilization method? So obviously autoclaving is carried out at degrees Celsius for 15 minutes for 15 psi. It is also called as steam under pressure. So autoclave is a moist heat sterilization. Pasteurization is two types: HTST high temperature short term and LTLT low temperature long term. HTST is also called as flash method, which is carried out at 72 degrees Celsius for 15 seconds, while LTLT low temperature long term is carried out at 62.8 degrees Celsius for 30 minutes. So pasteurization is also not the right answer over here. Filtration, of course, membrane filters are used with a pore size of 0.45 micrometer. So of course the answer cannot be filtration as well. Lyophilization is a freeze drying technique. So we have different lyophilizers used, we have different bulking agents used. So lyophilization is the right answer. It is not a method of sterilization. Next, the causative agent for mycobacteria. So mycobacteria species are detected by Zeal-Nielsen staining or acid-fast staining, which uses carbol-fuchsin as acid alcohol as a decolorizer, heat as a mordant, right? And secondary stain, maybe have different types of staining techniques like methylene blue, malachite green, and all different type of stain. So the right answer is Mycobacterium tuberculosis. Moving on, which antibiotic inhibits cell wall? Sulfonamides inhibit dihydropteroid synthase, right? While trimethoprim inhibits dihydrofolate reductase. Tetracycline inhibits 30S ribosomal subunit for protein synthesis. Erythromycin is a kind of macrolide and it inhibits 50S ribosomal subunit. Penicillin inhibits cross-linking via acting on the penicillin binding protein PBP and inhibiting transpeptidation. So it inhibits transpeptidation of NAMNAC, N-acetylmuramic acid, and acetylglucosamine, and thereby inhibits the cross-linking, and then transpeptidation is being inhibited. The process of conversion of sugar into alcohol by yeast is known as fermentation. By fermentation, you convert sugar into alcohols using a suitable bacteria or suitable yeast like Saccharomyces cerevisiae. It is a baker's yeast or a brewer's yeast, correct? Next, which of the following is a DNA virus? I want you to tell me the answer. I will eliminate one for you. HIV is a retrovirus. Correct? Let me know in the comment section, is it influenza, hepatitis, or rabies? The immune response mediated by antibodies. So, antibody mediated response is B cell mediated response, and B cell mediated response is known as humoral immunity. Moving on, which of the following is a live attenuated vaccine? So, when I talk about live attenuated vaccine, hepatitis B is a killed vaccine, influenza is a killed vaccine, pneumococcal is a subunit or a killed vaccine, but MMR, measles, mumps, rubella, is a live attenuated viral vaccine. So, the right answer is option number A. The enzyme reverse transcriptase is associated with obviously the bacteria is associated with a lot of other DNA gyrase and all. Fungi is associated with mycolic acid, squalene epoxidase. Protozoas are associated with heme polymerase, but viruses are affected by reverse transcriptase. That is why most of the drugs acting on antiviral are nucleoside reverse transcriptase inhibitors like acyclovir, ganciclovir, valciclovir, famciclovir, penciclovir, or non-nucleoside reverse transcriptase inhibitors like loviride, delavirine. Then, which of the following is a nitrogen fixation bacteria? So, Pseudomonas aeruginosa is a hospital-acquired community infection bacteria, which is nosocomial infection. E. coli is again a bacteria that causes urinary tract infection and GI disturbances. Similar with Staphylococcus aureus, which is a gram-positive bacteria. But Rhizobium species is a kind of nitrogen fixation bacteria. The process of killing microorganisms by heat is called as sterilization. Disinfection means you don't heat it. You usually use disinfectants, mild disinfectants or phenolic compounds, chloroxylenol and all, like Dettol, Savlon. Antisepsis is you prevent septic, but sterilization may you use dry heat, moist heat, radiation, or filtration in a methods of sterilization. Which of the following is a fungal infection? Typhoid is caused by Salmonella typhi, which is a bacterial infection. Malaria is caused by Plasmodium, so it is a protozoal infection. Female Anopheles, Plasmodium ovale. Tuberculosis is caused by Mycobacterium tuberculosis, a bacteria which is causing tuberculosis, and Candidiasis is a bacteria. It is a fungal infection which is caused due to Candida albicans and other species. So it also shows cross sensitivity with tetracyclines, the antibiotics. The study of fungi is called as obviously mycology. Virology is study of virus, protozoa, and bacteria is for bacteria. Which of the following inhibits protein synthesis? So, as mentioned above, cephalosporin cell wall, penicillin cell wall, sulfonamides DNA inhibition or dihydropteroid synthase inhibition. But tetracycline inhibit third ES ribosomal subunit, thereby inhibit the protein synthesis. The process of removing microorganism from a liquid passing through a filter. So pasteurization may though it is for milk, but it is not a membrane, it is at temperature. Autoclaving also there is a machine autoclave. Right? Filtration may you do that using a membrane filter of a definite pore size. Then we have which of the following is a gram negative. So E. coli is an example of gram negative, rest all are gram positive bacteria. The immune system that produces antibodies, B lymphocytes. The B lymphocytes basically produce the antibodies. Which one of the following is a DNA vaccine? You will let me know the answer. MMR is not a DNA vaccine, for example. Influenza vaccine is also not a DNA vaccine. Is it human papilloma virus? Let me know in the comment section how many people of you are confused between hepatitis and HPV. Let me know how many of you can give the right answer. The process by which a bacteria acquires a new genetic material. So transfection is a process whereby that is found in eukaryotes. Transduction is a process which is carried out in viruses. Transformation is carried out in bacteria. Conjugation means the fusion of cytoplasmic bridges. So the right answer is all of the above. Then the which of the following microscope is useful for where the light focuses on the specimen? So the right answer is lens. So in a condenser lens, the light rays focus on the specimen. You have different magnification power, resolving power, right? Limit of resolution. So you have different magnification power lenses. So 40x, 100x, 100x is cedar wood oil immersion lens. Then the term bacteriophage refers to a virus that can infect a bacteria is called as a bacteriophage. Then the which of the following is a gram positive bacteria? So E. coli is gram negative, Pseudomonas is gram negative, Salmonella typhi is gram negative. So the right answer is Staphylococcus aureus. Moving on, which of the following is used for Mycobacterium tuberculosis? Acid fast staining, I've already mentioned. We have Fontana staining for spirochete bacteria. We have Indian ink staining for capsule and all. And so similarly, the right answer is acid fast staining. Then, which of the following are the primary components of a cell wall? So, cell wall of any bacteria is made up of peptidoglycan. In gram positive, the peptidoglycan is thick. In gram negative, the peptidoglycan is thinner. Lipopolysaccharides are a part of gram negative bacteria, but the prominent portion of both the bacteria, positive as well as negative, is peptidoglycan layer. Finally, the endotoxins. So, in parenteral testing and in pharmaceutics, we usually do endotoxin tests by LAL, Limulus amebocyte lysium. We perform pyrogen testing because these endotoxin may cause pyrogenicity in fever-inducing organisms. So, they are present in the cell wall of gram-negative bacteria in the lipopolysaccharide. So the right answer is gram-negative bacteria. They are present in the cell wall of gram-negative bacteria with the lipopolysaccharides. So that's it for today's video. In the next video, we will be covering about the top 25 MCQs for biochemistry. So stay tuned and do like, share, subscribe my channel for more such upcoming videos.